If we start as hydrocarbons as the simplest organic molecules, characteristic chemical groups can replace one or more hydrogen atom bonded to the carbon skeleton of biological organisms. And these sort of Lego pieces of biology, these are what we refer to as functional groups. They're going to affect molecular function through their direct involvement in chemical reactions. Now, there are hundreds, probably, functional groups that exist. You are expected to know 7 key functional groups that are the most important to the chemistry of life. These are the same 7 that you should have learned in previous biology courses, but just in case, we'll look at them. Okay, first off, we have hydroxyl, or the OH group. The hydroxyl is a hydrogen atom that forms a polar covalent bond with an oxygen atom, which then forms a polar covalent bond with the carbon skeleton. Okay, we get the general idea. So because of these polar covalent bonds, hydroxyl groups increase solubility, and they are going to make these things polar. Okay, so organic compounds with the hydroxyl groups are going to be referred to as alcohols, and you'll see that in their ending, like OL, ethanol, methanol, that kind of thing. A carbonyl group consists of an oxygen atom joined to the carbon skeleton by a double bond. Where it is in the atom will determine whether it's if it's called a ketone or an aldehyde. You don't care about that unless you take organic chemistry. But carbonyl groups, these ones are majorly structural. They don't tend to alter the polarity all of that much, but they do have an effect on shape and properties. So isomerism, which we'll get to in a little bit, carbonyl groups can have a major impact there. Carboxyl groups. Carboxyl group is a C, a carbon atom with a double bond to an oxygen and a single bond to another OH group. Okay, compounds with carboxyl groups are referred to as carboxylic acids, because if you look really carefully, you will see that a carboxyl group is able to give up protons. So because it releases protons, it can make things acidic. Amino groups, which is a nitrogen, which is bonded to 2 hydrogen atoms. Okay, the amino group, you'll notice, is able to absorb protons, which makes it the opposite of the carboxyl group and makes amines or amino groups, makes those basic. Okay, so remember, anything that releases protons is an acid, anything that absorbs protons is a base. Sulfhydryl groups, this is a sulfur that is bonded to a hydrogen. It's like a hydroxyl group, but with sulfur. Organic molecules that have these are called thiols, but you'll probably never see that again outside of this. Sulfhydryl groups interact to help stabilize the structure of proteins in the amino acid cysteine, cysteine. That's the only time we're going to deal with the sulfhydryl groups. So sulfhydryl groups stabilize the three-dimensional shape of a protein using the amino acid cysteine. That will come back. Phosphate groups, which is a phosphorus atom bonded to 4 oxygens with various relationships there. One of them is double bonded, some of them are charged. You get the idea. You don't really care about that. But phosphate groups are anions with a 2 negative charge. And then one function of a phosphate group is to transfer energy between organic molecules. So we see this in things like ATP, for example. Adenosine triphosphate has 3 phosphate groups. Methyl groups, the methyl group is a carbon bonded to 3 hydrogens. We can see that relationship here. The methyl group is going to be attached in place of the various structures. The methyl groups become extremely important in complex genetics and epigenetics. So methyl groups are able to silence genes and alter gene structures. So when we get to genetics, we will talk more about the methyl group. But if you look for an example down here below, estradiol, the hormone that makes mammals female, and testosterone, the hormone that makes mammals male, the difference is that attached methyl group. So because the methyl group attaches here, that alters all of the other bonds within that structure. So really the addition of one functional group is the only thing that's different between females and males on a hormone level. When you then begin applying these things, like I said, I call them Legos because they are. You can assemble these in all sorts of shapes. Okay, we can see a phosphate group here, we can see an amino group here, sulfhydryl group, you got a hydroxyl, you got carboxyl, you got carbonyl, all of them are here. These things can be mixed and matched to make all of the different things that we consider living beings. One of the key phrases you're going to hear over and over is that shape determines function. So that is true on a molecular level as well as on the broader biological level. Isomers are compounds that have the same molecular formula but different structures and are therefore different in their chemical properties. Isomers might be structural, like you can see over here. Structural isomers, pentane and 2-methylbutane, the atomic formula is exactly the same, but they are structurally different and then therefore functionally different. You might have what's so-called cis-trans isomers, aka geometric isomers. That's all about how the molecules are arranged around a double bond, which is where something like carbonyl might matter. And then you also have enantiomers, which are sort of mirror images of each other. Looking at your left hand and your right hand, that is going to be an enantiomer. They are the same, but you can't overlap them to make that same structure. So enantiomers are a form of this. It's an asymmetrical carbon. Okay, even subtle structural differences can have important functional components. Morphine, for example, is an effective pain drug because it has the same shape as the binding site of the natural endorphins, those natural pain relievers. But sometimes that structure can be very problematic. Okay, that enantiomer, if it goes to the left or if it goes to the right, that's what's so-called chirality, only one shape may be useful biologically, whereas the other shape may be useless or indeed harmful. There was a morning sickness drug called thalidomide, and if you had one chirality, one enantiomer of it, it stopped morning sickness. But if you had the other enantiomer, it caused major birth defects in the babies. So definitely something that we need to keep in mind. Remember, for the most part in biology, we're going to only really be looking at covalent bonding because ionic bonding doesn't really matter within aqueous solutions because ions dissociate in water. So covalent bonding is really going to be how these things come together. In biology, overall, we really have two processes that will make and break these things. You'll have the dehydration reactions. The dehydration reactions are going to be bringing these molecules together. And then you'll have hydrolysis, which will be breaking these molecules apart. There will be fancy names depending on what it's breaking up and putting together, but this is that real base idea. You're going to have monomers, these repeating subunits that will be bonded together to form the polymers through dehydration synthesis. Okay, you're going to be removing water to make a bond, and you'll be adding water to break a bond. So in sugar, for example, the OH and the H are pulled out, leaving a glycosidic linkage behind. That is the name of the covalent bond via dehydration that occurs within sugars. So a lot of it comes down to vocabulary, but how do you form molecules in biology? By adding and removing water. That is going to be the making and breaking. Hydrolysis, water splits. Dehydration, pull water out to make a bond. That really applies to anything in biology, and digestion is no different. Okay, if you have enzymes are able to break down molecules, are able to sort of alter chemical reactions. So enzymes that do hydrolysis for carbohydrates, okay, salivary amylase, pancreatic amylase, all of these things, they're all using hydrolysis. Pepsin, trypsin, they're all using hydrolysis to break down a protein. Okay, enzymes are specific. They will only be breaking down, doing hydrolysis on one particular type of thing, only carbohydrates, proteins, whatever, but it is functionally the same. You're adding water to break a bond, you're removing water to make a bond. In our post-exam unit on anatomy, we will go into more details about how this works, but you should get a rough idea from this. An animal's diet must satisfy three nutritional needs: chemical energy, for cellular processes and all those kind of things, metabolism basically, the organic building blocks for macromolecules, and essential nutrients. Okay, chemical energy is going to come from the diet converted to ATP, fairly straightforward. Okay, organic building blocks, you're going to digest the food that you eat and turn them into new things. But essential nutrients are slightly different. And essential nutrients must be obtained from an animal's diet. Okay, that is going to include things like essential amino acids, essential fatty acids, but it's also going to incorporate things like minerals and vitamins. Okay, minerals are so-called cofactors. These are going to be non-organic molecules. Basically, hey, look, it's on the periodic table, and they are going to be cofactors because they help enzymes function, so they factor within that structure. Whereas vitamins are non-proteinous organic things, vitamin B3, vitamin D, vitamin A, vitamin K, all of those kind of things. Okay, now, essential nutrients are going to be different from organism to organism. Humans, for example, we have an essential nutrient of vitamin C because we cannot produce vitamin C, so we have to eat it. And if we don't get it, we have scurvy. Okay, whereas, say, chimpanzees never lost the ability to produce vitamin C, so they don't get scurvy. They don't have that as an essential nutrient. Now, that will also impact you on a broader biological structure. Malnutrition, for example, is if you get the wrong balance of those essential nutrients. That could be undernutrition, where you're starvation. It could be overnutrition, where you're dealing with obesity. Or it could be more specific than that. Within malnutrition, for example, we have marasmus and kwashiorkor. Marasmus is a form of severe malnutrition and can occur to anyone that is dealing with malnutrition, but typically affects children. Marasmus occurrence increases prior to age 1, and we'll get to see sort of that broader structure there. So marasmus, the child on the right, for example, is suffering from marasmus. The child on the left is suffering from a different disease called kwashiorkor. Kwashiorkor is a protein deficiency. The child is getting adequate nutrients, it's getting adequate energy intake, but the problem is that the diet is very high in sugars and fatty things. Now, it's not like the child's eating candy. They're probably relying upon things like rice and corn, which are very common in food aid because they're cheap. So this child looks like he might be obese, but that's not his stomach. That is his liver. Okay, because they are eating these things that are protein deficient, they're dealing with edema and muscular wasting and this buildup of fat in the liver. And so it's a form of malnutrition because the diet is not appropriate. Those broader chemical elements are cycled among the abiotic and the biotic components of an ecosystem. Photosynthetic and chemosynthetic organisms assimilate inorganic elements from the air and soil and water. So plants, for example, are capable of what's called biosequestration of carbon. They're going to take inorganic carbon in the air, CO2, and fix it as organic compounds like sugar. Okay, so because they're locking it away using biological processes and it's not cycling through the atmosphere, we call it biosequestration. Now, residence time is how long is that going to be locked away? Okay, we're dealing with that with carbon right now because we had biosequestration of carbon for centuries and millennia, but now we're digging up all of those carbon-based molecules as fossil fuels, burning them and putting them back in the atmosphere faster than biological processes can lock them away again. So we're dealing with an increase in CO2. These things, these biogeochemical cycles, it's not just carbon, nitrogen and sulfur and phosphorus. All of these things have biogeochemical cycles that we're expected to have some basic familiarity with. You'll go into significantly more detail on biogeochemical cycles if you take environmental science, and we also will come back to these when we get to ecology and the end of the course. But sort of just get a general idea. The balance between inorganic forms and organic forms, locking them away and cycling them through, that is a major component of the balance of nature and the balance of living things. At a more basic level, when you're looking at that balance, we have equilibrium and we have homeostasis. Chemical equilibrium is when the rate of formation of products is the same as the rate of breakdown of products. Okay, so we don't typically have true equilibrium in biology. We have what's called dynamic equilibrium. We're going to constantly have these things moving back and forth, and that dynamic equilibrium will mean that there is no net change in amounts. If you take chemistry, we're talking about equilibrium as K. Okay, so in the example here, we have the fish. You end up with 5 fish on each side. It balances out. K is equal to 1. Neither side is favored, and you're going to have equilibrium. That relates to the biological processes like homeostasis. Equilibrium is closely linked to the concept of homeostasis, which refers to the body's ability to maintain a stable internal environment. Homeostasis is essential for the optimal functioning of cells and tissues and organisms, allowing them to carry out their biological functions efficiently, whereas equilibrium is a key component ensuring that vital variables such as temperature and pH and blood pressure and hormones are kept within a very narrow and optimal range. So understanding components of these structures can be incredibly important, but we need to realize that equilibrium while it's fundamental, looking at that balance, okay, relies upon things like feedback loops and everything else. Ultimately, we are looking at organic nature of living beings, and are organic because we are carbon-based. Why is carbon such a central component to this? Well, carbon is tetravalent. It's able to make 4 bonds in all sorts of different directions, like we can see here, and that is going to enable carbon to form the base components of our carbon skeletons and our organic molecules. By attaching different atoms to that in different structures and forming these monomers and polymers and macromolecules, we get those base building blocks of life. So all those cells are between 70 and 95% water. The rest consists mostly of these carbon-based compounds. Okay, macromolecules are going to vary in cells of the same individual, but overall we have 4 major groupings. You have the proteins, you have the fats, you have the sugars or the carbohydrates, and you have the nucleic acids like DNA and RNA. Okay, we'll get more into that as we go into the next unit, but you'll notice here that this broad relationship here, or this schnapps, okay, carbon, hydrogen, nitrogen, and oxygen are in all of these macromolecules, depending on the structure, but they don't always use them in equal levels. Okay, carbohydrates, for example, that one is going to use carbon, hydrogen, and oxygen in roughly a 1 to 2 to 1 relationship, whereas fatty acids will use carbon and hydrogen and oxygen, not in a 1 to 2 to 1 relationship. Amino acids will always have nitrogen, okay, and amino acids can also have sulfur. Nucleic acids will always have nitrogen, but will also have phosphorus. So we can look at the balance of CH and O to tell the difference between a lipid and a carbohydrate. We can look at the presence of sulfur to identify a protein, and we can look at the presence of phosphorus to identify a DNA or RNA.